that has to leave, feel free. And the brainstorming, we're just going to brainstorm the basic structure of how to develop this collaboration more. So guys, we were actually going to have like a really cool presentation with Angelica. But she's going to give like a 30-second overview, and then we're going to have like a whole nother series, I think, with Angelica and Daniel, who actually have, I think from our last like smaller discussion, a great similarity in terms of your research question, what you're doing. But yeah, could you give like a 30-second?
Yeah, so the 30-second version, I'm not even going to write anything down, I'm not even going to stand up. So Rose told you what graphs are. You can pick random graphs. So you do that, you don't get to choose just any edges you want. You have your set of vertices, and then you look at each edge independently, and you flip a coin, and then if the coin comes up heads, then you put that edge in. And like maybe your coin is weighted, so you can have whatever probability you want, but it's the same probability for all of the edges. So you can figure out properties of random graphs. They basically kind of look a certain way, and that way is not what your brain looks like. Your brain has more structure. I guess if you model your brain as a graph, there are like clusters that are highly connected that are then connected to each other through through other hubs. I guess I think is basically how it goes, and that's not what random graphs look like. They're so in order to get the same so the way your brain looks, it makes it easy to get paths from between different points in your brain are pretty short. In order to get past that short in a random graph, you have to have a lot more connections, and so that's like so somehow your brain is more efficient than a random graph. There are like diseases and stuff that affect your brain, like Alzheimer's or whatever. I guess it looks like, from what I can tell, people think that like those make your brain look a little bit more like a random graph. So you're sort of losing some of this efficiency that's in your brain, and like moving more towards a random graph, which is not good. So the idea that we had that we were talking about was, so Sarah has these these neurons that are like, there's the healthy version and then the weird version. And it would be really cool if we could figure out a random, so some sort of a random model for, I guess, trees with some kind of a geometric structure, so that it's modeling these neurons well, so that like we can see that whatever the thing is that's happening when you move from the healthy one to the unhealthy one, or the one with a knockout, it's like moving more towards what you would see in a random model of that. So that's the short version.
So what is tree with geometric?
I have no idea.
Okay.
That is what would be cool to figure out. But like, so I mean, it would be like a tree with some sort of extra structure that's telling you something about like angles and maybe lengths of edges and stuff.
Yeah. So obviously we're not talking about topology really here anymore.
Yeah,
about
we
graph
move
theory
more into
and
like geometry. Yeah. But
that's still great and cool.
Astrology.
It's not a bad idea.
Just a question to people who know about graph theory. As far as I understand, graph theory is encoding who is connected to whom, but nothing about the distance between the things, whereas in the brain, somehow the distance is important in some way. So, am I right, or is there something I'm missing?
You are right to a certain extent, and there are obviously people here who know more about graph theory than I do.
I'd say, like, as far as I'm concerned, you're completely correct.
Yeah.
Okay, wanted to make sure.
I do think there are versions of graph theory that work with, for example, a weighted graph, where the weights would represent like how long the edges were. But that's...
Wait, what?
Can't you not reuse labels in a weighted graph, or is that a value graph?
No,
that's really different.
But weighted will be how well you are.
Labels is Jan's claim.
Weighted will be more, to me, like how well two nodes are connected, like how thick that connection or edges rather than...
But I mean, you can have the weights be represent whatever you want.
Oh,
right?
it could be a distance.
They could represent.
Right, yeah, right.
I think physical distance is important. That's the speed that information can travel down an axon, and that's more affected by things that are somewhat binary, like whether the axon is covering the mile sheet or not. So it could make the analysis more simple if you have just fast and just slow instead of the whole spectrum.
So then in that case we would have like, you would have, yeah, you're really having three things, right? You have not connected, you have connected by a slow thing, and connected by a fast thing. That would be it.
I have a question. question. So how does topology treat the morphology of a cell?
I don't really know. I think a
I don't think
lot
anybody
of,
knows.
yeah, a lot of the questions that Sarah is bringing up, in terms of studying the morphology of the cell, like, if I have two branches going off, what's the angle between them? Those aren't topology questions. Those are geometry
Right.
questions.
Those are questions that I can already calculate like trivially from the graphs that I have.
Yeah, the reason I ask about that is because, so I was thinking, so we are all, all the brains are wired differently, right? But we carry out same normal functions. I mean, if we are healthy, and the map that Sarah presented is a structure that varies from animal to animal, but they carry out the same function. So I was wondering if topology can answer the question that somehow even though the specific morphology of a cell is different, but somehow what comes out from the network is all the normal network reach to the same result, which is like normal result, normal function of the brain. But somehow, if the morphology is pathological, then what comes out from the network is something wrong, and maybe topologically different. Like if what comes out from one case is a sphere, comes out from the other case is a torus. Is that possible?
I don't know of any examples like that, but I imagine it would be possible. Yeah, that's a good question. Something that.
Then the question is how does topology model the morphology of a cell? How is that?
That is
how should I
yet to be done. It seems like it's like
Yeah.
a question, or yet to be studied.
I think if you wanna use like purely topology and not like consider the geometry, I think you would definitely want to be looking more at the network of all the cells than at one individual cell, because I think one individual cell is just gonna you can think about shrinking it back to so it's basically even I think the pathological cells are all gonna be. They're all going to look topologically the same because you can shrink them all down to a point.
Okay, so I think that the interesting thing for me between the morphology of individual cells and something that's actually applicable to topology is just like changing that morphology in a way that we can think about with graph theory or with various other sort of like statistical methods of just quantifying them, changes how they integrate into a network. And that's sort of what I was trying to get at by showing the retina circuit and the cortical circuit. In the retina, it's incredibly clear what happens when you change the morphology of a cell, how it integrates into that network, because it just can't receive stimulus now from part of the area that it's supposed to be receiving stimulus from. But in the cortex, we don't really know what changing the morphology of a cell does to how it integrates into a network. So taking those changes and being able to somehow characterize what that means for how it connects with the rest of the network is, I think, the interesting question. But we don't know the answer at the cell, and so we can't model it at the network level either.
But if we treat like each branching point as a vertex or something, yeah, then it doesn't shrink to one point.
So if I have a tree, topologically it's a point. So in graph theory and math, if I have a graph and I just have like a bunch of branches, it doesn't matter how long they are, we can just keep going. And it can have how many branches I want, I can have infinitely many branches, it doesn't matter. This, as long as I don't have a branch that like moves back around and starts back where I started, it's still topologically just a point. Yes, I can just take all of these branches and like squish them all back down to where I started.
So the confusion here is, so like a tree and a point are the same as topological spaces, but definitely not the same as graphs, right?
Yeah. So you would need tools from graph theory or from geometry to study that, not from topology. But it's still very interesting. So. Yeah. No, go ahead.
Can you do anything with hierarchical graphs? Like, say, a neuron with multiple branchiness is one thing with properties that are invariant between all of its components, but then that's connected to a larger network that also is composed of these little graphs that have their own.
So you're
sets
saying
of
like you have a
parameters.
big graph and then you have lots of little subgraphs?
Yeah.
Yeah, I mean, I don't see, well, I mean, so I guess it would depend on what, if your subgraphs and your big graph have topologically
So
interesting
is there any concept of coarse graining in graph? Something which might go like I have a huge network, but then I take these kind of look say maybe I put them in one big node, and then this which has more sub nodes. So can I coarse grain? Like in physics, you do these things all the time to coarse grain, but can I do that in graph theory, which might be related in this sense that if you can define layers of how intense your graph is, how many nodes, and is anything like that done anywhere in maths?
I don't know, but it sounds interesting. It sounds like a thing you could do. Probably what you would want.
I mean, of course you can do it if you define it right.
Well, sure. Yeah.
Yeah, I mean, you could, so I don't know if anyone is doing this at all, but you could just like quotient out whatever graph pieces that you
like look for parts of your graph that are like sufficiently highly connected or whatever, and then like shrink those all to a point. Something like that.
Something like that, yeah.
Yeah, I don't know of anything that's been.
But this
done
sounds
with
like
that,
the
but
sort
that
of thing that
sounds-
someone's probably
Yeah.
already done. Like it reminds me of hierarchically high topology spaces, and that's just like in our particular niche.
Yeah.
Yeah. It's also a little bit like what I was, that's kind of what you're doing when you add the simplicial complex. So you,
That's a good point.
yeah, so
Yeah,
like if
yeah.
the idea is that I have this graph, and then if, all 10 of these vertices are all pairwise connected, I'm going to add a 10 simplex there, and topologically that turns
That's a good
that
point.
piece into a point. So that's kind of what that is doing too.
Yeah.
I just think is there any way we can access your PowerPoint?
Oh, Sarah's PowerPoint?
Yeah, Sarah's
Yeah,
PowerPoint.
Oh, yeah, sure.
I feel like for system quality, you could, I gotta look at what the question is asking. But
I think what we're going to do is actually convert each of these presentations into like a small written thing, so that you can sort of like look at it and maybe break it up. Like some of the concepts that Rose said kind of went over my head just because I need like longer time to process. So it'll be on paper and you can sort of look at that. And Sarah's
Yeah, and
presentation.
I can certainly include like the one most informative figure as well in that, which is just the two diagrams themselves. I mean, like in this data set, I have like 750 cells or something, so those are just examples, but they're still informative.
And my other question was, I guess, whoever is the topology expert, I've been reading about like encoding the information that's contained in networks with sheaves. So far, I've got that some networks can be them, some can't. But I haven't figured out what information you get from the sheaf.
Yeah,
The sheaves.
sheaves are really complicated.
I don't know a whole lot about sheaves except for the
Yeah,
general
I
intuition
know.
that you can encode pretty much anything with them. So it's probably worth looking into more.
Can I ask very quickly where you were reading about this?
Classic.
So everything in there is discrete, right? So they're taking, so I'm sure it mentions categories if it mentions sheaves. And so you're defining your pre-sheaf from a discrete category, right?
I think so.
Yeah, so there aren't any like spaces with infinite numbers of points, right, that you're looking at.
No, I mean, he gave an example that is like a network of grades that are shared between middle school, high school,
Okay.
colleges.
Yeah. So I haven't read that, but I'm going to take a stab and say that probably what he's doing there is looking at something called the constant sheaf. And there's the constant sheaf can be used as sort of like really a calculational tool to calculate homology theories. And so it's it's a nice formal language because it allows you to do things very, very crisply. But on the other hand, you can also calculate those same homology groups using combinatorial methods, a bit more like what Rose was talking about. And so, there's sort of, I think, my guess would be that what you're doing in there is probably the same mathematics, just in slightly different language.
Yeah. I mean, he does talk about like torch rips and stuff, but if it's I gotta figure out if it's exactly the same as the sheet. Yeah.
Like to get back to what Angelica was mentioning in the beginning about why the mutant neurons might be forming a different branching pattern. And so there, how did you choose the particular protein to knock out? What was known about that in terms of molecular function or whatever?
So we don't, there are many known molecules that mediate the ability to recognize and be repelled by sister neurites, so just neurites from the same neuron, dendrites from the same neuron. REM2 isn't one of those. I was interested in it initially because it is an experience-dependent regulator of dendritic complexity, or at least at the time it was an activity-dependent regulator of complexity. So presumably, my visual cortex is being bombarded with different amounts of activity all the time, depending on whether I'm getting light input or it's dark, or there are patterns in my environment. But somehow my neurons maintain their shape more or less, and it's true in mice, and as far as I know it's true in humans. So somehow there's an activity-dependent regulation of this, such that neurons develop once there is light in an animal's environment, but they aren't constantly changing. So I was interested in understanding sort of the flexibility and stability of that system for this particular project. But this particular gene is a strong inhibitor of CAMK2, if anyone is aware of that molecule, which is sort of a master regulator for a lot of things in cells. So I'm telling you one story, but this particular protein does a ton of other things as well.
Neuroscience sounds hard. I'm glad I did math.
Well, to me, math sounds hard. I'm neither a neuroscientist, neither math, but I kind of understand neuroscience much easier than math. A quick question to the neuroscientist. So from what I understood today, especially when we were doing that simplicial thing, like, or even the random network, having too much network is also not, it's not efficient. Of course, not connected is nothing. So when we are developing, my naive idea is that actually nodes are not connected and you then start connecting. But does the opposite also happen very frequently, where things are already super connected with everything and then you kind of choose what to cut?
Yes, absolutely.
Are there examples, or is both happening all the time? Is one happening more?
So actually,
Okay, you should, no, go ahead. You do more than observation.
yeah, so it's something that's going on all the time in the brain. It depends on the area. So in the cortex, the rate at which the connections between cells are changing is much slower. And it's only like 5% of the connections that will change. But in other areas like the hippocampus, which is involved in memory, you actually get really rapid changes in the connections between cells. So basically all of the connections are erased within about a month, and new connections are formed.
Okay.
Yeah.
Okay, okay.
And how the cell chooses which new cell to connect to also varies depending on the region that you're in. So you could be, it seems like in the hippocampus, the new connections are almost random, although could be directed in some way. But in different areas of cortex, and especially in really young animals, the connections appear to be informed by the correlated activity of the two cells that are connected. So if they're more correlated, then they're more likely to have a connection.
So in a way, if I could somehow map out exact location of my connection, every new year I'm a new person. Revise. Maybe I'm maintaining the same
5 minutes.
Or every whatever minutes. Okay, I had no idea that. Okay. Fine. I didn't know it was that fast.
Yeah. It is much slower in some areas though. So in the visual cortex, where most of my work has been done, there's very little turnover in adult. As far as addition and subtraction of synapses, the connections can get stronger or weaker, and you can see that more frequently, but it's sort of very highly motile during development, and then it solidifies more or less, at least in the rodent. Of course, it's really hard to study this in well, especially in humans and even a non-human primate, so it's not as well known there. But in the mouse, it's pretty stable in visual cortex. as you can imagine, there's not a lot of changes in how we're low-level processing vision in our lives.
And these numbers change a little bit depending on your doctor too.
And which place of the brain it is.
Sure.
So I guess before, some more people depart, just really quickly, some boring stuff. How should we structure like the actual events themselves so that it like this makes all of you salivate? Like, what worked with this structure? What should we change? Because I also love the brainstorming component, and I want to develop that more because I think that's like this stuff was like so amazing just to see brainstorming. So what feedback do you guys have in terms of like when we do future ones?
Well, one thing is not 3 people in one
Yeah,
session.
yeah, good point. Okay, only 2 people at the very most. Okay, what other stuff? Any other feedback on like how to make this mixture better?
I mean, I think since this is the first one, it's a little bit hard to do, but I think it would be nice in the future if there were pairs of people going who had like constructed a more like related content. So like we were both just sort of like, the original intent was like I was going to introduce neuroscience like as a field. So a lot of my stuff is like, this is the brain, it has this many neurons, like this is the synapse, so like we just got rid of all of that and I had to jump into stuff that like probably came at you kind of fast. So I think it would be good if there was some more combined content, like say like place cells came up, if someone wanted to talk about place cells, get like a dot lab person and like they could talk about a related topic so that it was more uniform. But for the first one, like yeah, you just sort of have to intro a bunch of stuff all at once.
Yeah, we could definitely be more focused, I think. We could even like, this is a particular study or a particular technique that we want to like read all together, and let's, this is a study about the visual cortex, so Sarah's going to give everybody a presentation about things, basic things that she knows about the visual cortex, and then, they are using this particular topological technique, so I'll do some research about it and talk to you guys about that technique, and
Yeah, I think that would be really fruitful.
And then the other 2, oh sorry.
Just out of curiosity, has anyone in here read the paper that came out I think over a year ago now, 11 Dimensions in the Brain? I think people.
I've heard a lot about it.
I heard that in
Yeah.
string theory there is a 11 dimension in brain.
Yeah. Well, I gathered they used topology somehow to write that title. And if there's anyone here who has a decent understanding of that paper, I would love to hear more about it.
The Paul Miller's group, I guess they're all gone now. I'm sure one of them could do it, because Paul Miller has like a physics background too, and so we could just ask him, and he would do it probably.
Yeah, I would be, because it's. I have to say, topology is like a really, really big field, and like Rose really only touched on like the very basics of it. And it's very, very hard, I think, for a lot of the math people in the room to know what areas of topology are actually going to be useful to the neuroscientists.
Yeah.
And so it might be really good for us to have an example of how topology has been used productively in the past.
Yeah, I think that would also benefit many, maybe not many, maybe I'm just flattering myself, but it would be very helpful for me to be able to read about it a little bit before coming to a talk about it, just to sort of increase my basically non-existent knowledge.
So like a journal club, like we do also a few journal clubs on like a few key papers that highlight the connection between these 2 fields.
Yeah, just an example of how they've been combined in the past.
Okay.
There aren't that many, that would be fantastic.
Yeah. And in terms of the brainstorming, so option 1 is what we're doing here, where we have an hour presentation and then maybe 30 minutes plus of brainstorming right after, while it's still fresh in our minds. Option 2 is like, we just had the presentation, we convert that into a document, people read it before, and then we have a meeting number 2 a week later or something, where it's just brainstorm. Which of those 2 options do you think is better for brainstorming?
I think it's better than
Talk first, and then right after do what we're doing right now. Nice. Yeah,
I think you're more likely to get participation that way.
I agree. I think so. Yeah, I don't know.
Just to be realistic.
Yeah. I don't know if people get exhausted or not. It's like an hour and a half, but like I'm pumped. So like for me, it's definitely doable.
We can have each portion be shorter.
Wait, Eric, is this replacing GSS or is
No.
it in
No.
addition to GSS?
No,
Okay.
this is just this week.
Yeah,
Since we didn't have a speaker for this
I
week,
see.
and I said,
No, GSS is
We
going
haven't
to keep
opened.
going same as always. We just didn't have a GSS this week.
Okay.
I think I prefer this style to the journal club style. I think for a journal club is, if you haven't read a paper, it's hard to get to the meaty stuff of the paper within half an hour or 1 hour. Right?
Unless we're going to send it out ahead of time, like significantly, which would be good.
I
I would need more presentation to get it like 100% sure.
No, it makes sense. So it makes sense that we do the presentation, but then we embed actual works that have been done, with just like a quick synopsis so people can go look at it and stuff like that. And then, yeah, we could do like one that kind of focuses, but still the discussion based on 11 dimensions. In fact, like we could go to the Computational Neuroscience Journal Club and invade that and just have like, one of them do it. And that might be really cool too, right?
Right now,
Yeah,
here's this paper.
Yeah, no, I feel like they would.
Perhaps some useful takeaway if you're a neuroscientist is that topology allows you to put the word dimension in your title. Pretty much no matter how you use topology, that can be done. So if you want to write a paper about how the brain is 12-dimensional, I'm sure you can swing it.
I want to see a paper the brain is infinite dimensional. That would be so much more satisfying.
You could probably swing that too.
It'd be infinite dimensional sounds weirder than it
It depends on how you define dimension, right?
is a vector space. It's an
I
infinite
see.
dimensional vector space.
Okay.
Over a finite field.
Yeah, people would definitely say the name of that paper aloud many times in mixed company.
Yeah.
That's smart.
So, before maybe we even conclude, since this looks like a more core group, maybe we can just quickly just say who we are and what field we are and what you're interested in, just so like, that might be cool. Because part of what I think is really cool is if we socialize a little bit too, maybe all go party and stuff and make these like bonds so that we can really create collaborative ideas afterwards. So maybe we can just quickly do that in a circle. So I'm Vivek Anand, I'm a postdoc in neuroscience in the Ashton Grable Spatial Orientation Lab. And I'm running experiments too, where I strap humans to a machine that falls over, you're blindfolded, and use a joystick to balance yourself. So if you want to experience that experiment, and Lila's running some too, contact us. You can come by our lab, and we'll run you, and you'll get paid $10 an hour or something like that. Cool. And then, then we'll
But can we come by just to like try it
You can just
out?
come by, You can just come by just to check it out too. But it'd be great if we can run you too.
What's the tax rate on that with the new plan?
You owe him $1.
They pay you $10 an hour, you pay the government $20 an hour.
So Josh and Angelica and I are all students here working under Ruth Cheney. So she's a geometric group theorist, which is I'm not even sure if it's a subfield of topology. It gets grouped into topology, but
We will accept you into topology.
yeah. Thank you. We really appreciate it.
You're half of the topology seminar.
Yeah, but
This semester they're the whole seminar.
Anyway, so we all work with the same professor, and basically we study groups which are algebraic, and we use geometry to study them. And I do a lot of with, not necessarily simplicial complexes, but things like that. So that's why I thought that was a good place to start. I
Yeah,
kind
I'm
of already
Josh.
introduced
Basically
Josh, but-
that, yeah. Well, I work with the same advisor.
We're going this way, or?
Yeah, let's do it. Seems like it.
For sure. I'm Noah. I'm an undergraduate. I do theoretical work in Eve Marder's lab. Primarily I do computational modeling and I'm trying to understand how degenerate sort of features of